Okay, so hello again, everyone. Welcome to our webinar. Today we will discuss how neurons can be used as biodigital sensors. For that, today I'm joined by my colleagues, Serge Roux, who is our product director, who has a high experience in mechanical engineer and industrial design, who is today leading our product development. We also have Hélène Gauthier, our biopharma scientist who has a background in neurosciences and is currently our expert in electrophysiology. And today she's leading the R&D development of PNS and pain platforms here at NETRI. And we're also joined by Rania Talbi, who's our data scientist with experience in data engineer and machine learning. So just to give you a quick overview of today's program, we'll first start to discuss how characterizing sensory neurons is important and can be used as a universal biodigital sensors to explore such applications, such PNS applications. We'll then go through how we'll create digital signatures, and we'll of course finish with a quick Q&A session. So in case if you have any questions, do not hesitate to write them in the chat of this webinar. So before we start discussing the subject of today's webinar, just wanted to quickly show you how today in NETRI, the idea behind our devices. So currently, as everyone knows, today when we want to screen different drugs, usually what we do, we would use different types of cells mixed up in a well of a 96 plate. But this is not relevant for the human anatomy, hence we have natural compartmentalization. The first one being the one that is obvious. It's the compartmentalization between the external environment and the internal environment through the skin. So with different types of stimuli, such as, for example, a pressure, the sense, or the heat or the cold. And of course, we have another type of compartmentalization, which is an internal one between different organs and also between different systems, for example, the vascular and the nervous system. And so the idea behind our devices is to recreate this physiological compartmentalization using our microfluidic devices. As you can see on the right, we have a screenshot of our individual chip, which is actually our fundamental device, the dual link, where you have 3 channels with 2 main channels and one in the middle that will create a type of barrier that will not enable fluids to go through the 2 adjacent channels. And so in those devices, you can, for example, culture one type of cells in, for example, a first channel on the right, for example, such cell types such as neurons, and in another channel, the target cells or the target organ. And so this will enable to have a model of innervation. And of course, we have a new device that we have pre-launched this summer. And if you're interested in it, you can watch our webinar number 4 on our YouTube page. It was around our multi-fluidics line with duplex, where you can have a central well that can culture, where you can culture 2D and 3D cultures. And this central well is adjacent to two channels that are related to a bottom reservoir that can act and mimic vascularization. And you have the bottom reservoir is, of course, separated from the top middle well by a porous membrane. So you can use the system to test different types of compounds, or even to nourish if you have in the middle well, you can have different types of organoids and test different types of compounds that can be brought by the vascular system, for example. And so the idea with our devices is that we today have a unique selling solution which first mimics the physiological compartmentalization that you do not have in classic 96 well plates. Second of all, because our chips are made from a breathable or a special type of silicone, which enables exchanges of oxygen, so you don't need to add any pumps or additional devices to make sure that your cultures stay alive. And also, we have our devices, they have the same dimensions as classic 96 well plates. And so our devices are compatible with most lab equipment. And so, as you can see in this particular new bento, on our chips, you can see at the bottom, underneath the chips, you have different electrodes. And so the idea is to be able to capture the electrophysiological responses of neurons. And that's where comes the idea to use neurons as biodigital sensors. In fact, neurons can react to different types of stimuli, whether it be external or internal, and they also interconnect different types of organs. And that way, we can assess and study different specific phenotypic readouts in a non-invasive way. And so the idea is that according to a type of stimuli, the neurons will respond in different ways. And this electrophysiological response is what we call digital signatures. And this is what we today are striving to create. It's a digital library using reference compounds. And to then be able to test target drugs or target compounds, and to be able to predict their, for example, efficacy or toxicity in reference to these compounds that we have in our library, and we have characterized. To sum up what we have today at NETRI, so if you're interested in our devices, we can first, obviously, we can sell unique devices if you want to test directly our products in your lab. Of course, we also propose screening. We have developed a couple of different platforms for screening of drugs. We have today the idea of neurons as a sensor, and we are also launching as an early access platforms for neuromuscular junction and vascular skin. And of course, if you have other interests, we also can co-develop new platforms. If you're interested, for example, in studying blood-brain barrier or circulating immune systems, we can, of course, co-develop new platforms with. And so on the right, you can see different types of our current and future applications that we can use, for example, oncology, toxicology, and of course, the subject of our today webinar, PNS and different PNS applications. And so on this note, I will let my colleague Serge take the lead on PNS applications.
Okay, hi everyone. So the fundamental idea that our system allows is to basically consider that everything you feel is actually based on your neurons. Whether it's pain, whether it's pleasure, whether it's aches or itches or scratches, everything boils down to the signals that are being fed into your brain. So the whole idea is, if you think about it, a neuron is not, you can consider it not necessarily as a neuron, but basically as a sensor that senses everything for you. And therefore, if you take that to its logical conclusion, you can, by analyzing what your natural sensors are actually experiencing, it gives you it would give you a really good information on the different compounds or treatments or different stimuli that you apply to different organs of your body. So our generic approach to PNS is that you can basically, if you culture neurons on one side, you can put on the other side stimuli that will get encoded by your neurons. And because the idea is that if you have electrophysiology, you can effectively digitally encode the information that is there already digitally produced by neurons, and that you can do so with neurons alone. That can bring you a great deal. You can do it by taking supernatants, and alone and compounds, or supernatants that basically has processed compounds from organs, and you can go all the way, if necessary, to co-cultures. So that's kind of our generic approach. But in order to do that, you have to have an effective sensor. So let's talk about the neuron as an actual sensor. So first, you need some software and hardware platform to be able to receive the informations of ones and zeros that neurons produce. So first, the first thing you have is that's the compartmentalization that Nasiba was talking about allows you to put somas in one side and neurites on the other side, which is basically what happens in your body when you have your spine and your organs on the other side, whether it be your skin or your gut or what have you. So this replicates the natural organization in your body and allows you to potentially test compounds or stimuli on the end effectors, which is where that happens usually instead of directly into your spine. The other thing, to note is that our fluidic isolation here means that whatever you put inside this compartment on the right, which we call channel 3, will not actually get processed by the other side on channel 1. So you actually are mimic the physiological behavior around neurons. Then comes the MEA layer, which is the multi-electrode array layer that goes under this compartmentalization and allows you to get readouts on any of the channels, and more importantly, in the microchannels in between the channels. So you can see here a culture of sensory neurons where you have your somas on this side that have proliferated and projected to the other side, nice and orderly through our 3-micron microchannels here, which means that somas stay on one side, but the axons grow to the other side. And that allows to do fluidic compartmentalization. And we have ultrasensitive electrodes here in the microchannels that basically pick up the signal of what's going on in channel 3 going over to channel 1. And that's kind of the key benefit of doing this for PNS, since information travels towards the soma. We have an integrated platform with our partner. Our MEA sheets are actually manufactured specifically for us by our partner, Axion BioSystems, and we use Maestro, which is the world-leading electrophysiology readouts, electrophysiology recording device, with which we have paired a specialized software to extract compartmentalized information and then process it through our internal database, which we call Data Link. So we've put together a hardware and a software platform. So you can see here how we have our software uplink, which basically processes the information that is taken generically from the Axion BioSystems system and chops up the information into different channels, whether it be channel 1, the microchannels, or channel 3 on the other side, and you get this information in this kind of layout, where basically you can get every single chip has all its metrics, but all its configuration of channels. So you can see the mean firing rate of channel 1, or of channel 2, or channel 3, and so on and so forth, which allows you to, depending on your application, to look at different places in the geography of your chip and draw meaningful results, whether it is that you've stimulated on one side, on the other, on both, in the middle, and so on and so forth. So this is basically our extraction. I just talked about it before. Your Maestro allows you, with Axion's Axis Navigator, allows extraction with uplink, and then we do storage and analysis on our Data Link database. So that's for the hardware and software side. Now, I'm going to let my colleague Helen take over, because basically, if you have a software and a hardware platform, you need the right kind of sensor characterization on the other side to make sure neurons do act as sensors.
Thank you. Yes, exactly. You need cells in order to have all of this working. So what we're doing at NETRI is really we are adapting cell supplier protocols specifically to our chips and making sure that they are developing properly in terms of maturation of some marker expression. For example, here you have the example of TRPV1 or substance P that are characteristic, specific of sensory neurons. So here's an example of what we do in order to validate cells in our devices. But we look really what is most important is their electrophysiological activity and making sure that they are they develop according to what we call our internal standards. And what we make sure before doing any kind of treatment is to quality control our cells by MEA recording, a basal MEA recording that will allow us after to compare. So here you have some quality control is based on a minimum of baseline activity and also a certain number of active electrodes before we can consider that the chip is okay to be tested. So we have a little tool that calculates the mean, so you can either refer to a global database. So we are building database specific to each cell type, but you can also do it just locally with for your experiment to make sure that you don't have outliers that should not be treated either. So here in the example, in red you have either a chip that fell because there was not enough active electrode or a chip that fell because activity was too low to start with. If you look at the bottom, the evolution of the culture, this each dot is a chip that has been tested in our devices. So we can also look back and differentiate if a new batch of cells, a new lot, is responding how it should. You also have next to it plotted all the MFR from chips that have been recorded, all our historical data on activity and showing that they are following a normal distribution in our devices. So it is essential for us to have this first baseline reference recording. So each compound or induced disease or induced injury, we can compare the electrophysiological activity in this altered state compared to the reference state and looking at percentage of changes within the same culture. Depending on how we are, the type of compound we are testing, we can either look at what we call either time response method. So we will do a recording before application of compound, and then at different time point after compound addition, we'll do follow-up recordings, and we can look at the changes as expressed percentage of change from the relative reference state. That can be a healthy culture, or for example, if we do look at temperature, it can be the reference temperature at 37 degrees. We can also look at how the platform, the culture, respond to specific agonists or compounds, and look at how what is the fold change from the baseline. So we will look this time at local local activity and look at the maximum of activity, these fold changes from the basal activity and the difference between our positive control, most of the time KCL, and the vehicle, and look at how the compound is positioning itself in this window, this assay window. If I go a little bit more into how does it look like in real, so this on the top left, you have one of the raster plots that Serge mentioned before, and compartmentalize. So here you have a color code with a channel 1, channel 2, and channel 3 in shade of blue, and the microchannels, the electrodes that are picking up the axonal activity, are either in yellow and red. And this is an example of a sequence of events that we will use to assess the response of the neuron. So here's our mechanosensing test that is done by an hyposmotic solution to stretch all the mechanoreceptors. You can see a clear response from the neuron, and especially it's clear as in microchannel, but also in the channels, despite the fact that we didn't detect as much activity in basal in the channels. So the quantification is done by looking at this as a maximum of the response and normalizing by the basal activity just before the addition of the test of either the vehicle, the hyposmotic solution, or the positive control KCL at the end. And we can express the data, for example, either as a fold change like this, or the percentage of our KCL response. Now I will present you a kind of overview of how you can use of application for the platform, and some applications that we already have evidence that you can use the platform for. So so either you can do protocols of time response or a fast response, depending on the type of agonist. So for example, if you want to test for ATP, it would be with a fast response protocol, or some protective effect or inflammation will be more tested with time response. And so you have example of the target or the known target, or if the known, I will say if you don't have a specific receptor, and I will show you example of compounds that we tested in those conditions. So, for example, if you want to test more, if you're interested in prurate or itching, we've been able to show that the platform can answer to difference of temperature, but also responsive to, we can look at ATP induced response and how these sensory neurons are activated by it. Also, we can target pH or TRPA1 receptor with lactic acid. Similarly, the platform can also be used for a hot-cold effect. So here's an example. So you still have the example of the temperature and how we can differentiate, have different electrophysiological signature depending if the cells are at low or at a high temperature compared to a 37 baseline. And those changes are really dynamic. If you look at the bottom graph, you can see that when you set the temperature, a few seconds later, you will see the change in activity, and the change of activity will be a bit slower because it takes a bit longer for the hardware to go down in temperature. We started to have evidence that we can also differentiate and have a really specific profile when we apply an agonist for cold receptors such as WS12 testing. Regarding PEN, so the same platform of sensory neurons on a compartmentalized MEA can be used for inflammation of pain management. So by using, for example, time response protocol, being able to show that a difference in the response to temperature when our culture are treated via vehicle or treated with a known compound for inflammatory pathways such as TNF-alpha. In order to define some window of assay window and making sure that the platform can be used to test analgesic, we compare and we tested lidocaine, showing that we do have sometimes sodium voltage-dependent sodium channels that can be blocked by lidocaine. Other example of what can be done in terms of pain field. So paclitaxel is still a kind of not all the targets have been known as well labeled as broadband, that there's multiple targets, and you can assess and try to understand which one and how the cell are responding to this compound. So in the example, we had we tested sequential addition of paclitaxel and shows that the response is more subtle and different compared to, for example, some of the previous slides with the ATP, which was a really big. So we start to have evidence that depending on the compounds that we're adding, we're getting different response. And that we can then trying to understand a bit more of those responses to build a liberate different signature of those responses. The last application for this platform is it could be used to understand a bit more IBD or the gut-brain axis by using either secretion, metabolite, neurotransmitter, or cytokines inputs, so we've been able to show that LPS added on its own, so that it will be inflammatory, will increase the electrophysiological activity. And we've been able to show also with the platforms that you can distinguish a molecule that will have protective effects, such as the propionate, as shown in the example here, that we protect against the LPS effect by adding propionate in the cultures.
Just to reinforce, in all of those cases, the sensors were the same, and the compounds were added in channel 3. Right, so this basically shows that we have a very targeted, like we're actually targeting the receptors on the neurites on the right hand side without and basically monitoring the signal going up towards the somas.
Yeah. But I think that's where I'm
okay. So,
letting you the floor if you want.
Let me go back one. So basically, we talked. This is basically all kinds of applications that are just using the sensitivity of our sensors on a basic metric like MFR or WMFR, where it becomes more where things take a really and this is already quite novel and interesting, but where we can kind of push the envelope of it further is by trying to start and be specific. Okay, so it just so happens that, as a lot of you know who are working with MEA, there's an enormous amount of metrics that we actually get access to every time we do recording. We've been only showing here MFR, right? So what we have is a combination of two things. We have a lot of average metrics, and those are really those that basically come into play in our case in PNS, because there are no networked activities not in sensory neurons. So we're looking at the average metrics on the left hand here, but we also have our combination of channels. Okay, so when you think about this, you basically have a large matrix of information. So if you say, for instance, that you have all the metrics in the channel 1 configuration here, and this is just illustrative, just to give the sense this is not real data, and this is kind of relative data for every single one of the metrics on channel 1. Well, you do have that for every single configuration. So in fact, you have that, which is basically which gives you much more information. So not all of it is going to be useful in every case, but you get access to this information and you can process it in bulk to see which part of this information is relevant. Okay, so when we saw this, we said, well, actually, this kind of looks like a QR code. Okay, where one of these little squares would be the mean firing rate, right? But all the others in the mean firing rate in one of the channels, in channel 3, for instance, or in the microchannels in the middle. So you basically you can look at this as a digital signature between a reference state which can be a healthy culture, or it could be a diseased culture, and an altered state, which is trying to make it healthy again, or basically inducing a certain pain, or inducing or coming back to a baseline of things like this. So basically, it's a differential digital signature. So this is conceptual, but what does that actually mean when you start to try and put it into practice? So, yeah.
I can, try to, well, so we started to see, can we see differences? Is this, if we look at more than one metric and different condition, can we start to build a digital signature or see differences between two conditions? So here we took two sets of data. One chips that were challenged with temperature, and with or without a test compound. And by doing a PCA, by comparing two different metrics, we can see that we're starting to have a space in two dimensions where the two conditions started to be separated. It means that by taking more metrics into account, we can discriminate with more specificity.
And that's basically being simple and just looking at two different metrics that create a specific pattern. But if you want to kind of generalize this and make it more of a general process that you can apply across a range of reference compounds, you can basically start to look at things like this.
That are more, n-dimensional. So here we tested, we wanted to look at different edges of our assets. We define a asset window by choosing, testing our the workflow and all the analysis that were done with test compounds. So in this example, we use veratridine that it's really known to induce burst-like activities that are really well defined. Again, we use lidocaine as a control that you can that is used also in clinic to have a reference for an analgesic, a powerful analgesic effect created in this system. And after running a PCA, principal component analysis, and so Rania might correct me later, by removing some of the metrics that are correlated together, we can extract a matrix of similarity from that. Here, what is presented, it was done on the 5 most relevant metric, and we look at the distance, comparing the effect of one compound and how distance, the set of 5 metric is from this test compound.
So you basically get a 5-dimensional vector, and you look at the distance between those 5-dimensional vectors, which basically in this case is 5. It could be more, it could be less, and it's actually, so the process actually filters through all the configuration of the QR code to identify in the different configuration, the configurations that are most discriminative, more that are the most discriminative, and then within that, you can actually look at the metrics that are most discriminative inside the configuration that are the most discriminative. Okay, which is kind of the power of the system, is that it gives you all those configurations to actually start looking for the discrimination. And as Hélène was saying, what you get is on the PCA plot over there is basically a playing field where you've defined the corners of your playing field. And if you start looking at a compound, you can start looking at whether or not it starts to look similar to one or to the other, or to a whole gamut of reference compounds to come. And that's basically the slide that we want to leave you with. Is the idea is, if you have a certain number of reference compounds for a particular application, okay, and those compounds can be simple agonists, but they can also be known compounds that are on the market. Very interestingly, if the information is available, compounds that have failed to go to market, and allow you to create a digital library, which means that if you have a target compound that you want to test in reference to those, you can start seeing matches and correlations, the distance the distances between those n-dimensional vectors to inform your your choices for screening your compounds. And to go even further, if you were starting to look at the digital signature of a particular condition, say ALS or say some other, then you would be able to actually potentially also diagnose certain patterns with patient samples. And we're not talking about right now, obviously, but basically this is where this is all kind of going towards. And I think that's basically, that concludes kind of the panorama of how of our journey to turn neurons into digital sensors. Yeah.
Yeah. So thank you, Serge. Thank you, Helen. So thank you, everyone, for listening. And so we can start our Q&A session. So if you have any questions, do not hesitate to send the questions in the chat. Okay. So first, we have a question, maybe more for Serge. So can this approach work without a full library of reference compounds?
So yes, this is basically an incremental process, right? You can start creating a local digital library with reference compounds that are known to you, that you know that are the kind of like that create your sandbox, right? And then this becomes a local comparison. But as the database grows, you can start getting out of your own reference compound and start to explore other people, like the other compounds in the digital library.
Okay, and so can this digital signature approach be used today?
So yes, today we already have a workflow, a pipeline workflow that we can use that internally to actually run basically what you've seen, but based on compounds of your choosing.
Okay.
That's a question for
Helen. We have a question finally for Helen. How do you do mechanical stimulation in channel 3?
So we're adding the IPOS osmotic solution directly in the channel via the inlets. And we record, and the response that is recorded is really what is traveling through the microchannels that we are looking at to really mimic this, that it's the periphery that we are testing. And
so the mechanical nature is actually done through osmotic channels?
Yes.
Right? So it's not a mechanical as in pressing on it. Although it's PDMS, so... Yes.
Okay. We have another question, maybe more for Rania. How is similarity measured between compounds?
Yes, if we can go back to slide 37, I think. So basically, as Serge mentioned, similarity is measured over an n-dimensional vector that we build for each compound based on the metrics we compute using our using Axis, our partner tool. And we don't use all the metrics. We basically use methods to identify which metrics are the most important, which are included in measuring the similarity. Techniques for measuring importance of these metrics include information gain, weights obtained from a PCA analysis, or LDA. So once we selected the most important metrics, we use distances to measure the similarity between different compounds. In this figure, for instance, we use the cosine similarity, but it could definitely be something like a weighted distance, which takes into consideration the importance of each metric at a site. So I hope it's clear.
I hope we answered your question. So today we talked about PNS applications mainly, but can this approach be extended to other applications such as CNS?
Yes. The answer is yes. So right now, neuron as a sensor, we're looking at it where the sensor is a sensory neuron because we're looking at PNS, right? But if you go to CNS, you can choose a healthy, standard, calibrated CNS cell type, which would be.
I would go for
glutamatergic neurons. Thank you. And then basically use glutamatergic neurons as the recipients of neural activity on, say you put glutamatergic neurons in channel 1, you can have them be the recipients of a neurological activity that happens in channel 3, and in channel 3 you could have, well, other gluta, or you could have a mixed population of an area of the brain, or you could have diseased neurons, or you could have whatever, and then basically record the activity of channel 3 inside this activity of channel 1, which in effect makes gluta become your CNS sensors. We do have there's a publication coming that should give you some information about that.
Okay, thank you, Serge. Another question for Rania. What is the typical pipeline applied for compound similarity evaluation?
Basically, slide 37 is their reference for this kind of question, I guess. So basically, our analytical pipeline starts with using our partner tool Axion for spike detection, noise filtering, burst detection, and so on. Then, along with our own tool adapted to our compartmentalized devices, we calculate a set of metrics, which we then pass through the pipeline to carry out filtering of outliers of two metrics that are too correlated with each other. We also carry PCA for visualization purposes. We also assess the importance of the metrics generated by our tool Uplink into discriminating different compounds using various techniques that I mentioned earlier. We carry clustering to identify common sets of compounds that behave similarly, and we generate figures, for instance, such as similarity measurement between different compounds. So we can also consider supervised tasks such as toxicity prediction, but this is in another context.
Okay, thank you, Rania. Maybe we can have one last question for Helen. Is this approach limited to axial sensory neurons?
Yeah, not only, as also Serge says that you could use for CNS application with another type of neurons, for example, glutamatergic neurons. We did tested different. There is a few data that are not coming from axon cells, but from other sensory neurons that we are testing, because we would like really to be able to recommend the best model depending on the readouts and the needs.
Okay. Actually, we have another interesting question for Helen. All this is supposed to work only if the plated neurons in channel 1 grow neurites with similar branching in channel 3. We all know this is not the case. How we calibrate between neurites with and without branching?
I'm not sure. I'm not sure I understand well the question here. We're mostly interested when we talk about sensory neurons, is the activity, we're sure it's axonal because we stimulate in channel 3 and we record on the journey from channel 3 to channel 1. So even if there is some projections that are, it's biology, so we don't always defined, but everything that goes through and long enough will be axonal and can be tested by adding compounds or stimulating channel 3. Hope
we answered your question.
Yeah.
Makes sense. Okay, so I think we will end our Q&A session here today. In case we Okay. In case if we hadn't the time to answer some of your questions, we are going to the Neuroscience 2024 conference organized by SFN in Chicago. So in case you're attending too, do not hesitate to come and meet us at our booth, number 1372. And of course, everything that we discussed today, you can find it in our application note that we have released on our with our partner Axion BioSystems on our resources webpage. And if you download this application note, you can also have you have a code to save 10% on our NeuroFluidics MEA line if you're interested in our MEA devices. And of course, if you want to review or share this webinar with your colleagues, this webinar will be available on replay in on our YouTube channel, of course. And so you can watch it in replay tomorrow. So again, thank you, everyone. We're happy to answer your questions and present our new advances in neurons as biodigital sensors. And thank you, everyone, and have a nice day. Bye.
Thank you.
Thank you.